Hello guys, hope you all are doing well. Today we are here with a chapter namely reproduction in plants. In this part we are going to cover asexual reproduction. Right. So before going to start a chapter we must know what is reproduction, right? So if we say on a basic, reproduction is the process by which a living organism or a creature produces a new individual. So here a question arises, why to produce a new individual? So the answer to this question is that for the continuity of a species. For just imagine our ancestors or the humans in the early Stone Age wouldn't reproduce, so we won't be here. So that's the main purpose of reproduction, and here also comes in catch that each and every genes or next generation is better than before or have superior qualities most of the time. As you can get it, for example, you can take the humans of the early Stone Age and humans of the 20th century. There is a huge difference between them, right? So that's the improvement of the species. So let's move on to the chapter. Yeah, I need to explain that this chapter is for class 8 ICSE students. So moving forward, we have asexual reproduction. As you can see this here in the mind map, there are majorly 7 types, where these 3 are the previously read. I mean, you have been reading this in the class 7. This is something new, and all these 3 are also new to us. So before starting, let me show you the names. First is budding, second is binary fission, third is fragmentation, fourth is spore formation, fifth is vegetative propagation, sixth is artificial vegetative propagation, and seventh is tissue culture. So let's first read these 3. So let's move on to the budding. Wait a second. Yeah. So you can see the definition here. Let me highlight it for a better understanding. The process of producing new organism from their parent is called reproduction. The new organism is not exactly same but somewhat similar to their parents. That's what I was saying. That the new organism which is produced is not same as their ancestors or parents. It is somewhat better, or sometimes it also gets worse, but most of the time it's improves. So there are on average 2 types, asexual reproduction and sexual reproduction. So in this part we are going to cover asexual reproduction. So what is asexual reproduction? Asexual reproduction is the simplest method of reproduction in that take place in organisms whose body have simpler structure. So asexual reproduction takes place in the body of organisms which have simpler structure, that is, they don't have sex cells or gametes. So what are these sex cells? So these are in the higher organisms. Moving forward, there are majorly 4 types, that is binary fission, budding, fragmentation, spore formation. So first we are going to start with binary fission. In binary fission, binary means 2 and fission means division. So here you can see this parent. Let me zoom in for the better and a quick understanding. You can see here it is the parent cell or body which divides into 2 daughter cells. So what happens? The parent body divided itself into equal halves. Here you can see there is an equal division of both nucleus, this part which is surrounded with a blue thread-like structure, and the outer mass with green dots. You can see here that is the cytoplasm, and the matter which is enclosed in the blue or purple thread-like structure, that is your nucleus, right? So there is an equal division of that too. So what happens? The parent body divided itself, giving you can say that the parent body kind of extends, or does not have longer existence after it divides into two daughter cells. Right, let me read you the definition, then let's again move on to the diagram. It is a division of parent body into half to produce daughter cells. In this type of asexual reproduction, first the nucleus elongates and the division divides into two, and then cytoplasm splits across the middle to form two daughter cells. Each grows as a separate individual. So what is binary fission? That is a parent body divided into two parts, namely two daughter cells with equal division of cytoplasm and nucleus. The only thing that is here to remember is the name of the parent body, that is parent body, and the divided cell, which is daughter cells, and there is only division of two cells, not more than that or not more less than that. The body will divide into two, not three, not four, only two. That's why it is known as binary fission. Here it is also written 2. Right. There is also a part, I may think I have mentioned it here. Let me show you. Yeah. Binary fission and multiple fission. So let me explain you the multiple fission, but it is not mentioned in our syllabus right now, so we can skip also, but let me explain you. In multiple fission, can you see, let me zoom in the diagram. I may assume you can see this darker portion or the outer wall of it. It becomes more thicker and acts as a cyst, kind of a thick wall, which prevents the interior, which is cytoplasm and nucleus, from the outer harsh conditions. When there are, just for an example, you can take when there is wind or storm in your locality or your area, your house works as this cyst and you are safe inside. So, in multiple fission, there is not division of two cells. There is a division of numerous cells which rapidly divides inside this wall, and after the favorable conditions has occurred or came, the wall will break and it will divide and then it will spread into the environment. So let's move on to the next part, which is budding. So let me read the definition first, then I will be explaining you with this diagram. In budding, an outgrowth called bud arises from the surface of the parent body. This bud grows bigger with the accumulation of cytoplasm and nucleus, and also divides and passes on to each end. The bud ultimately gets cut off from the parent body and leads an individual life. So what's the meaning of that? It's simple. If you can see the diagram, let me show you. Yeah. So here you can see it's a diagram of yeast reproducing. So this red part, I know this picture is kind of blur, but there's some issues in this with the quality. Let me continue with the chapter. Can you see this red dot type structure, which is nucleus? So here, this dark and light portion, the lighter portion is the cytoplasm, which is getting elongated. See, in the second part, this second picture of this, there's a small kind of acne or pimple type structure occurring in the cell body, that is your bud. In the third picture, you can see there is a division of nucleus with it. So what happens? It's not the equal division, it's a small part of it which gets elongated and divided. And when it is matured, in the third picture of it, you can see that it is almost matured, and in fourth picture it is mature. Then it gets separated and leads an individual life. The only difference which we are having from the binary fission and the budding is that in the binary fission there is no longer existence of the parent body which is divided into two daughter cells, but in budding the parent body is still alive. It was that simple. Let's move on to the next part, which is fragmentation. In this type of asexual reproduction, the filamentous body of lower organisms such as Spirochea breaks up into two or more fragments and grows as a new individual. So you can say that it's kind of similar to the binary fission, but a bit different, cause you can see the parent body here. Let me zoom in the picture. It divides into smaller fragments. You can say like cakes. I think you have eaten that 10 rupees or 20 rupees cakes which come in a packet of Britannia. So there are small pieces you can see. So imagine the cake as the parent body and small molds as the pieces of the cake. So it depends on the organism to organism that how many fragments on which it will be dividing. If it is large or bigger, it will divide into 4 or 5 fragments. It can also divide into 2 fragments, 1 fragment, and each and every fragment of it will be growing as a separate individual. I mean to say here is, in binary fission, there will be only division of 2. The parent body will divide into 2. But in fragmentation, the division will depend upon the type of the organism which is dividing. It can divide into 4, 5, 6, 7. It depends on the organism. So this was here up to the class 7th, which we are revising. Yeah, I am making some blunders while speaking. Let me continue. Spore formation. This is something which we are getting new in class 8. So let me start with it. Some organisms like bacteria, fungi, mushrooms reproduce asexually. I told you asexually means in which there is not the fusion of the sex cell or gametes by the process of spore formation. So you can say spore formation is kind of a process which some organisms such as bacteria, fungi, mushrooms use to reproduce asexually. That means by their own. To explain more about spore formation, we are going to talk about bread molds. So if you can recall, there are some kind of fungus when you leave your bread out in the kitchen slab or something like that. Like you are going to eat a tasty bread jam, you are ready to put the jam on, but your mom said to go out and bring some vegetables for her, and you forget it. And you are so lazy that you forget it, and after 2 days when you are back in the kitchen for eating another thing, you saw that the bread has got some molds on it. Let me show you the picture, that will be a bit easier for you all to understand. Just wait a second. Yep. If you can recall, its picture is something like this. There's a bit trouble here. Yeah, it's something like these type of structure occur on the bread. So these are the bread molds. Right. If you can recall, let's continue the chapter. It's a type of spore formation reproduction. So it also occurs in different it occurs in different type of organisms. Mold are type of fungus that reproduce asexually by spore formation. As we know, each spore contains nucleus matter and a small amount of cytoplasm enclosed within a tough outer wall called sporangium or spore sac. So here the important thing is this definition: spore sac or sporangium. So what are these spore sac and sporangium? In these, the cytoplasm or the nucleus matter is enclosed, just like and the cell wall. If you can recall, in plants, the cell wall acts as a thick wall which prevents or surrounds the nucleus and cytoplasm. Just same like that is the spore sac or sporangium. It contains nucleus matter and small amount of cytoplasm enclosed in it. They can resist unfavorable conditions. It's just like a wall I told you before. It will prevent it will act as a wall to prevent the cytoplasm or nucleus to get destroyed due to the harsh condition. When the sporangium is mature and finds suitable environment, it will burst and release spore. Each spore germinates and will give rise to new mold. So you can see the diagram here. Let me zoom in. Yeah. So this is your spore sac. You can see there sporangium. When it will protect the mold. I remember that what I told you before about the multiple fission. It's almost similar to this, in which the parent body develops a thick wall which is similar to the sporangium, and it will divide rapidly inside that. When a suitable condition arises, it will release spores in the environment. Each and every spore will form new molds. Let's move on to the next part, which is vegetative propagation. In vegetative propagation, we have 4 types: underground stem, aerial, by root, by leaf. In which we are going to start with underground stem. Underground stem also have 4 types: bulb, rhizomes, corm, tuber. Let me explain you all about that. So, what is vegetative propagation? Wait a second, there is a trouble here. Yeah, that's fine. The ability to produce new individuals through vegetative parts such as stem, root, leaf is known as vegetative propagation. So what are the vegetative parts? Vegetative parts means a part from where vegetables can grow in a plant. You know that the vegetables are kind of a storehouse for the food which are produced by the plant. So yeah, the parts where the plants preserve or set the food to be is the vegetative part of it, such as fruit, vegetables and these all, and which grow on the stem, root and leaves. It takes place in a plant which do not have seed or flower and helps in the sexual reproduction. So it's kinda acts like in a flower in the plants which do not have seeds or flowers. So natural vegetative propagation. Wait a second, there's a trouble here again. Yeah, natural vegetative propagation. There are three major types of natural vegetative propagation, namely not 3, 4. Yeah. Underground stem, by root, by aerial stem, by leaves. By underground stem. Stems such as bulb, rhizome, corm, tuber and runner help the plant to reproduce vegetatively under favorable conditions. So what is a bulb? First type. A bulb is has a small fleshy leaf called scales and a bud which grows into a new plant. So here's an example. First we need to read the examples, which are onion, garlic and tulip. So if you can see, it's an example of an onion where if you have ever cut or got in the kitchen to help your mother, you will see that when you are cutting an onion, there are different type of layers inside it. These layers are known as scales, and inside it, in the middle part, the last or the innermost layer, which is the bud, that is the only part which grows into a new plant. If you are having an old onion or onion which has not been used for a long time, you will see there is a outgrowth. That is this green part you can see on the top of each onion. It is having such a growth when you are not using it for a long time. So let's move on to the next, which is rhizomes. Rhizomes are the branched underground horizontally growing stem. They have distinct knots and interknots which are axillary buds. These axillary buds can grow into new plants. So what does that mean? First see the examples: ginger, turmeric and banana. So here horizontally means, let me show you by drawing it because it will make it easier for you to all to understand. Wait a sec. Yeah. Yeah, no, it's fine. So horizontally means, for example, take this is our ground. Most uneven ground ever. And don't mind my drawing cause it's really bad. This is our plant. Fine. These are the leaves of the plant. Yeah. Yeah, then. And these are the roots. So rhizomes will grow in the shoot of the plant, root of the plant, and it will grow horizontally. Horizontal means in this way, and vertical means in this way. See, so this is, just imagine this is our rhizome. So it will having it will be having distinct knots. If you have ever seen ginger, you will see there are kind of layers or scales. If you can see, there is kind of a line type structure at each end, something similar to this. See, if you can recall. So at each end, at internode there is a bud which grows into new plant. So for take an example, when a farmer grows an rhizome and he forgets to pluck the plant out and leaves it inside the moist soil for a long time, it will grow into a new plant because of the birds in the internodes. The main point you need all to remember is that the plant will grow only and only from the bud, not these internodes or these. You need to remember these where the bud is located, nodes and internodes. The main thing or the major thing that will turn into the new plant, that is bud. Next, move on to the corm. And corm is another type of underground stem that grows vertically. The axillary bud borne in the axil of scale leaves produce corm, which later grows into a new plant. So what does that mean? First, let me show you that picture which I drew before. The most beautiful picture ever. So here you can see our rhizome was growing horizontally. Let me denote it with H. And corm is similar, just almost similar to the rhizome, but the difference here is that it will be growing vertically. Means if it is smaller like this, it will grow into larger piece, but growing vertically, not horizontally. It will be also having knots and internodes here, and each internode will be bearing an axillary bud, which will further grow into a new plant. If it is left under the ground in a moist soil under the ground for a long time. Okay? So moving to the next part, which is wait a sec. Yeah, tuber. A tuber is a modified fleshy and slow and underground stem which contains food for the plant. It has many buds called eyes. Each eye grows into a new plant, example potato. So the tuber is also similar to the rhizome and corm, if you can relate with it. But the difference here is that it will be also growing, it doesn't matter it is going vertically, horizontally. The only thing that's different is tuber does not have any scales like this one and this one, corm and rhizome. It has distinct eyes, you can see like this part, it has eyes, and each and every eye can grow into an individual plant, such that if you forget to take it out, or a farmer who is growing that forgets to plug the plant out, it is inside the soil for a long time, it will be growing into let me put a different color here that will be able to display. Damn, I did something wrong. Wait a sec, please. Black. Yeah, fine. So each and every eye will grow into a new plant. Let me show you the diagram, cause my drawing will make you really confused. So let me open the diagram there. Yeah, you can see here in the picture of it is an example of a potato. There isn't any knots or internodes, just like this. You can see these knots, these lines on the ginger, or in the corm, these lines which act as the internode. It is having different kind of spots, just like an acne or pimples, which each and every these spots can grow into a new plant. It is kind of a small flowering part of it. Flowering, I'm saying, a small plant or a plantlet type of something similar to that. So the only difference between these three or four are bulb is having scales and a small bird inside it. Just imagine this picture. Second, rhizome, it grows. It is also, it's having internodes, knots and internodes, and will grow horizontally. Corm is also having knot internode, but it grows vertically. Tuber does not have any knot or internode. It has eyes. Each and every eye can grow into a new plant. Let's move on to the next, which is aerial stem. Runner and stolon. Wait a sec. Runner and stolon are creeping stem which produce root at the knot. The vertical stem arises at the knot, forming new plant at the intervals. For example, dovegrass, wool, and strawberry. So, what does that mean? If you can see here, in a plant, there is a small creeping part of it, which is almost lying on the ground, gets near to the ground and grows into a new daughter plant. So how does that happens? In a runner, these creeping plants have different knots and interknots which bear roots. You can say when this small part of the shoot or the stem goes near the soil, it will start producing root into the ground and to absorb mineral, and when it will be able to absorb the mineral from the soil, it will detach itself from the parent plant and lead its individual life. So let's move on to the next, which is by roots. The adventitious roots also help in the vegetative reproduction. They become fleshy and tuberous. They have adventitious bud which can reattach itself and grow into a new plant. So let me zoom in the picture. Yeah. So if you can see in this picture, it is also kind of, you can say, sweet potato, or swollen root, tuber. It is also written here, tuber. So the only difference which we are getting here is that the bud will grow from this green part, the stem, not from the root. You can say that when the shoot enters, before the division of the root, at the point where it's in the middle of the shoot and root, I mean stem and root, there will be the bud which will grow into a new plant. That was that simple. Let's move on to the next, which is by leaves. I think that this part is the most complicated for most of you all, but it's really simple if you can get it. Let me read it first. Leaf of some plants like Birophilium and Begonia produce adventitious bud on their margins. When the leaves fall on the moist soil, these buds develop into small plantlets. On separation from the leaf, these plants grow into an individual plant. The fleshy leaf of Birophilium provides food and water to a developing plantlet. So what happens? The leaves of some special plants such as Birophilium or Begonia, they bear small plantlets or small leaves on their main. You can say that the leaf of the plant acts as another plant, and it will be bearing small plantlets, as you can see in this picture. When it's completely matured, when there are enough small plantlets, it will fall off the main plant body and will be acting as a new plant when it falls on the moist soil. Then the question arises here that how will it get the nutrition, because it is not having any roots or something similar to that to have nutrition. So until and unless it produces its roots after falling on the soil, the nutrition and these plant hormones and these all will be provided by the main larger plant on which small plants are growing. You can see here the fleshy leaves of Birophilium provides food and water to the developing plantlet. So it will contain all the food and plant needed for the plantlets to grow until and unless it is completely prepared to grow into a new plant. Artificial vegetative propagation. So which we have learned before is vegetative propagation. In that, the major thing that happens is the plant reproduces from its vegetative part by their own, but in artificial vegetative propagation we need to do it. So we are done with the underground stem, aerial, then by root, by leaf. So moving forward to the artificial vegetative propagation, there are three types of artificial vegetative propagation. First is grafting, second is cutting, third is layering. Done. And last is the tissue culture. You can say that we are on the second last topic for this part. So let's move on to the artificial vegetative propagation. Some new plants are developed vegetatively by the human being to improve the variety of the plant. Such propagation is called artificial vegetative propagation. So what does that mean? It means that the plant which is developed by the human beings to get a superior quality of the plant or something better, that is called vegetative propagation. Let me read it again. Some new plants are developed vegetatively by human beings to improve the variety of the plant. Such propagation is called artificial vegetative propagation. Grafting. In grafting, two different plants are combined to obtain new plant with superior qualities. In this method, a stem cutting of one plant called chicane is attached and tied to rooted stem of another plant called stalk. The major thing you need to remember here is chicane and stalk. These two are the major parts to be remembered. After few days, due to the rapid cell division, the chicane and the stalk merge together and develop a new plant with improved qualities. As stalk is rooted, it provides nutrition to the chicane. Using this method, we can get different colors of rose plant in a single rose. So, let me show you the diagram for the better understanding. You can see this chicane. It is the shoot or the part of a plant which bears the leaves and other flower, leaves, fruit and these all. And stalk is the rooted part, which is you can say the stem or the part of the plant which is rooted. You can see it's in the second picture, it is combined, and in the third picture it is tied with a cloth or a rope for that. So when it is combined like this, there is a rapid cell division between them, which makes them combine. And the major function of the stalk is that it will provide food and nutrition to the chicane, whereas the chicane will be bearing the fruits and vegetables with the superior qualities, as both of them are having different qualities. As you can say something like that your father and mother are having different blood groups, such as your father is having A and your mother is having B. I'm not sure, I don't know about a lot about that, but you can, for example, I'm saying that your mother is having blood group B and your father is having blood group A. When you are there, you are produced, you are having the blood group AB, so you are having some superior qualities, right? So it is not, I don't mean that it's compulsory to happen or the formation of AB blood group by that, but you can say like that something, that the superior quality by combination of them. So let's move on to the next one, layering. In this method, a young healthy branch of a plant is bent and a part as it is covered under the soil in such a way that its terminal bud is exposed to air. After a few days, the root develops from the plant under the soil. The branch is then cut off from the main plant and planted it separately, which grows into a new individual. Such example is strawberry, lemon and jasmine. So it is the simple layering here is similar to the one which we have read before, runner or stolon. But the difference here is the runner and stolon grow by their own, but in this layering we need to put a healthy moist branch of a plant inside the soil. And the major thing we need to check here is the aerial or the part of the plant, such as where the leaves are bearing. If you can see the part after the layering of the branch, there is an small leaf type structure outside the ground. That is the major thing you need to remember. The terminal bud, you can see here, is exposed to air. The bud means the main plant which will help it to grow is outside in the air and the stem is inside it. After a long time such that about 1 month, the roots will develop which from the part which is under the ground, and then we need to cut the stem to make it separate. So this is a simple layering method. So let's move on to the next one, which is cutting. In this method of propagation, a new plant are produced by cutting the part of a stem of a healthy plant that has axillary bud. This stem cutting is then planted in a moist soil after a few days. The root develops from the portion embedded under the soil and a new leaf develops from the bud present in its upper end from the new plant. So what does that mean? Right, let me explain you. You can see in the diagram, this axillary bud or the upper part of the shoot is cut off from a mother plant or the parent plant and is then planted in a small pot. So it is simple. You can also do that by most of the plants have this property. You can also try that if you are having any plant. You can just cut off the healthy shoot which is bearing the bud, plant it into a new pot, and after a few days or months, when you are giving water, soil and all the nutrition that it needs, you will see the stem which you have planted inside will start to bear roots which will help in the growth of the plant. So here we are done with the artificial vegetative propagation, and the last part of this video is tissue culture. Not the least but the most important. So let's start. Tissue culture is a technique of raising new plant from a small portion of a plant tissue. So this is a major thing. The small portion you can read here, that means that, let me highlight this is the most important part, the small portion of the plant tissue. It means here you can say about some sort of small cells, a group of small cell, or something like that. Only a short part of a plant. You can say also some pieces of fragments or pieces of a leaf, something like that. Let's move on, read furthermore. Over a culture or synthetic medium under a sterilized condition. What is a sterilized condition here? What do they mean by sterilized condition? So a sterilized condition or a synthetic medium is the part which contains plant hormones or the nutrients that are important for a growth of a plant. So if you are having a doubt what are plant hormones, this is also important. What are plant hormones? So plant hormones are some hormones that help in the growth of you can say we all human eat Bonvita with milk, and these all Bonvita, Horlicks, Kellogg's. So these are something like the plant hormones for us, like hormones which help in the growth of us. Our body also have some natural hormone that is called growth hormone. So let's move on to the method. So let me give you a simple idea of it. We are taking a small piece of fragments of a leaf or a plant, giving it a sterilized medium which contains nutrition and plant hormones, and for its proper growth. It's not like you are taking a small part of a shoot or that you are planting it. You are taking it into a sterilized medium. So when you will be reading this method, you will get to understand. So let's read. These steps which are involved in the method of tissue culture are: first, a small piece of plant tissue is taken from a growing tip of a plant, such as tip of a shoot or root or shoot. So you can say we need the tip. The last part can be said as the top of the shoot or that. So second part, the tissue is then transferred to a test tube containing suitable nutrients medium under a sterilized condition. This medium contains plant hormones. So what I said before, this tissue is transferred to a test tube containing suitable nutrient and mediums. So it is not like planting it. We are taking it to a test tube containing suitable medium or hormones. Due to the presence of plant hormone, as I said before, division and forms undifferentiated mass of cell called callus. So you can say when we are putting it into a sterilized condition under test tube, it will form a mass of plant tissues called callus, which will further divide to form plantlets, and this plantlet is now prepared to get planted and develop into a new plant. These plantlets are transferred into a pot or soil growth for attain maturity. So yeah, here this part ends, and you can see let me explain you with the diagram cause it may help you. So we are taking the plant, cutting it into a small fragments, transferring it into a small test tube type structure containing plant hormones and nutrients. It will be forming callus, which will develop into plantlets, and then after that we are planting it into a pot. Right. So here the chapter ends. Thanks for watching, and if you are having any query, I will be pinning this mind map in the description, so you can check it out there, and this PDF also. Let me show you this, and this PDF also I will be pinning it into the description, so you can go and check it from there. Thanks for watching.